Hi, I'm Khalid, a member of the Neuroplicity team. Now, most people, when they think of neuroscience research, think of studying the brain, and specifically neurons. But there's much more to the brain and nervous system than just neurons. In fact, half the cells in the brain aren't neurons at all, they're glia. Today, we're going to take a look at some of the neuroscience research being done at Duke University on these important cells that neuroscientists are just beginning to understand.
Hello, my name is Cagla Eroglu, and I'm an assistant professor in Duke University Medical Center in the cell biology department. My lab is investigating how our brains form, particularly how the connections between neurons called synapses form during development, and how these synaptic structures are remodeled with learning or with disease. I'm particularly interested in how non-neuronal cells such as glia influence these processes.
Understanding synapses is the key to understanding Dr. Eroglu's research. So let's break it down for you. Synapses are the meeting points where neurons communicate with each other. The neurons aren't physically connected, though. Instead, there's a space between them, called the synaptic cleft, where chemicals are released by one neuron and detected by the next neuron. That's how one neuron sends information like, "Ow, this is hot," to the next one. It releases chemicals into the synaptic cleft. Those chemicals are then detected by the next neuron, which passes the information on to the next cell, and so on until your muscle is told to move your hand off the hot stove. Our brains are constantly making new synapses and losing others. The formation of new synapses is called synaptogenesis. Synaptogenesis is critical for brain development and is a process that researchers like Dr. Eroglu are working to understand. Dr. Cagla Eroglu and her lab study a special glial cell called astrocytes that play a key role in synaptogenesis.
Astrocytes actually make the majority of the cells in human brain, and unlike neurons, they do not have these action potentials where their communication is more enigmatic and less understood. So we've been investigating how astrocytes interact with neurons and how this interaction leads to formation or remodeling of synaptic circuits. Particularly, we're interested in secreted proteins that are coming from these cells that influence neuronal connectivity.
Astrocytes and other glia have been thought of as support cells in the brain for a long time. They hang out around neurons, secrete things that neurons need, and take up things that they don't need. But it wasn't really understood until recently just how much influence astrocytes have on neurons, and especially on synaptogenesis. Dr. Eroglu's experiments have shown that something astrocytes were secreting was actually telling the neurons to form more synapses. Dr. Eroglu and her team wanted to figure out what this secreted factor was. She hypothesized that it was a protein called heaven because 1. It's found in the same place as synapses, and 2. They already suspected that astrocytes, and not neurons, were the ones producing it. First, they used fluorescent microscopy to look at a mouse brain. When they found that there was an overlap between astrocyte cells and heaven, they concluded that astrocytes do indeed produce heaven. Next, to figure out if heaven has any effect on neurons, they grew neurons in a petri dish and added pure heaven protein. This caused neurons to make about 5 times more synapses compared to when no heaven was added. So it looked like heaven, made and secreted by astrocytes, was indeed causing the formation of new synapses between the neurons. But then, the researchers grew the neurons in a solution that other astrocytes had already been growing in. Since the astrocytes secrete heaven into the surrounding solution, the team figured that this would have the same effect as just adding heaven to the neuron petri dishes.
However, when it was in the full complement of the astrocyte secreted factors, it was no longer able to induce synaptogenesis. So this was very curious and suggested to us that there were other factors coming from astrocytes that masked or inhibited heaven's synaptogenic activity. That led us to identify Spark, which is, I would call it a cousin of heaven. So in the protein world, they are related.
Dr. Eroglu found that astrocytes secrete heaven, and heaven causes neurons to make more synapses. But when neurons are in a solution containing not just heaven, but everything that astrocytes secrete, then extra synapses don't form. This led Dr. Eroglu and her team to hypothesize that astrocytes might secrete another factor that competes with heaven's effects. She guessed that the other factor might be a protein called Spark, which actually looks quite a bit like heaven, which is why she called it a cousin. To explore this hunch, they went back to the petri dish. Neurons that were untreated were a control group and told the investigators how many synapses are normally found between neurons. In another dish, neurons were grown with purified heaven, and they formed way more synapses than untreated neurons. In the third dish, neurons were grown in purified spark, and here, about the same number of synapses formed as in the control group. In the final dish, neurons were grown with both heaven and spark, and also ended up making just as many synapses as the untreated neurons. These experiments showed that spark alone didn't prevent synapses from forming, but rather kept heaven in check, essentially blocking, or inhibiting, heaven's synaptogenic activity.
Spark, instead of being an inducer of synaptogenesis, had actually an opposing function. It inhibited synaptogenesis. And we found that when these proteins were not there, let's say when heaven is not there, there are less synapses, and when spark is not there, there are more synapses. So the significance of these findings were that astrocytes not only induced formation of synapses, they also provided factors that would, limit that activity, and that way they can more carefully control the number and the location of synapses that need to be formed.
By exploring the less understood cell types, astrocytes, Dr. Eroglu has shown that astrocytes directly secrete many things that can enhance or inhibit synaptogenesis. So, rather than being just passive support cells, astrocytes play a very active role in whether or not neurons form synapses with each other, and therefore how well they'll talk to each other. Dr. Eroglu and her lab have made many more discoveries like these, showing how important astrocytes are to our brain. For more information, visit Dr. Eroglu and her team online.